So, here's something worth thinking about as you settle in tonight. Right now, inside your skull, there's a 3-pound organ that's using about 20 watts of power, roughly the same as a dim light bulb. And it's doing something that no supercomputer on Earth can fully replicate. It's keeping you alive. It's processing the sound of my voice. It's pulling meaning from these words, converting vibrations in the air into language, into ideas, into images. And somewhere in the background, it's also regulating your heartbeat, monitoring your breathing, cataloging the events of your day, deciding what to remember and what to quietly let go of, all at once, all with the energy budget of a light bulb. That's your brain. And honestly, the more you learn about it, the more astonishing it gets. But here's the thing. For most of human history, nobody had any idea what the brain actually did. I mean that literally. For thousands of years, brilliant people looked at the human body, studied it, dissected it, wrote enormous volumes about it, and most of them thought the brain was basically irrelevant. A lump of tissue that maybe cooled the blood or produced mucus. The real action, they figured, was happening somewhere else entirely. Let's start there, because it's a wonderful story, and it tells you a lot about how hard it is to understand something when you don't even know what questions to ask. The ancient Egyptians were remarkable engineers and physicians in many ways. They performed surgeries, set broken bones, had pharmacological treatments that actually worked for certain conditions. And when they mummified their dead, which they did with extraordinary care, preserving organs they believed the deceased would need in the afterlife, they carefully removed the liver, the lungs, the stomach, the intestines, and placed them in special canopic jars, sacred vessels, protected for eternity. But the brain? They pulled it out through the nose with a hook and threw it away. Just discarded it. Because to them, the brain wasn't important. The heart was where you thought. The heart was where your soul lived. The heart was you. And they weren't alone in thinking this. This idea that the heart is the seat of consciousness, of emotion, of the self, persisted for an incredibly long time. We still carry echoes of it in our language, right? You learn things by heart. You speak from the heart. Someone breaks your heart. We say, "I knew it in my heart," when we mean something deeply felt and true. That's not just poetry. That's the residue of an ancient, deeply held belief that the heart was the organ of thought. Now, the earliest document we have that actually describes the brain in any medical detail is something called the Edwin Smith Papyrus, and it's fascinating. It dates to around 1600 BCE, though it's probably a copy of an even older text, maybe from 3000 BCE or so. It's named after the American antiquities dealer who bought it in Luxor in 1862. But the real author, the original author, is unknown. Some scholars have speculated it might trace back to Imhotep, the legendary Egyptian polymath who was architect, physician, and eventually deified as a god of medicine. We can't be sure of that, but whoever wrote it, they were observant in a way that feels startlingly modern. The papyrus describes 48 cases of traumatic injury, starting from the head and working down, and it's in the head injury cases where things get really interesting. The author describes patients with skull fractures, and they note, with remarkable precision, that damage to one side of the brain can cause problems on the opposite side of the body, Think about what that means. Somebody, roughly 4,000 years ago, watched a person with a wound on the left side of their skull lose the ability to move their right arm, and they wrote it down. They didn't fully understand why. They couldn't have. But they noticed. They documented the correlation between brain injury and bodily function, which means at least some ancient Egyptian physicians suspected the brain was more important than the official cultural story suggested. But that observation didn't take hold. It was a footnote, a curiosity. The heart-centered view dominated for millennia. Fast forward about 1,000 years to ancient Greece, and you get one of the great intellectual battles in the history of understanding the brain. On one side, you have Aristotle, one of the most influential thinkers who ever lived, a man whose ideas shaped Western thought for 2,000 years. And Aristotle was firmly, confidently, absolutely wrong about the brain. He believed the heart was the seat of intelligence and sensation. The brain, in his view, was basically a radiator. Its job was to cool the blood. He had reasons for thinking this. The heart is warm. It responds to emotion. It's clearly vital for life. The brain, by contrast, is cool to the touch. And when Aristotle poked at brain tissue during dissections, the animal didn't seem to feel pain. So he concluded it couldn't be the organ of sensation. On the other side of this debate was Hippocrates, or at least the tradition associated with him, since much of what we attribute to Hippocrates was probably written by his students and followers. The Hippocratic text called On the Sacred Disease, which is actually about epilepsy, makes an extraordinary claim for its time. It says, essentially, that the brain is the organ of thought, of sensation, of joy and sorrow and pain. That epilepsy is not a divine curse, but a disease of the brain. The author writes, and I'm paraphrasing here, that people ought to know that from the brain, and the brain alone, arise our pleasures, joys, laughter, and sorrows, and our tears. Through it we think, see, hear, and distinguish the ugly from the beautiful, the bad from the good. That's remarkable. That's someone in the 5th century BCE getting it essentially right. And yet, for centuries afterward, Aristotle's view held enormous sway, because Aristotle was Aristotle. His authority was almost impossible to argue against. Now, the person who really started to shift things, who built the first serious anatomical case for the brain as the command center of the body, was a physician named Galen. Claudius Galenus, born around 129 CE in Pergamon, which is in modern-day Turkey. Galen is one of the most important figures in the entire history of medicine, and his story is worth spending a little time with, because it tells you something about how knowledge actually advances messily, brilliantly, and with a lot of wrong turns mixed in with the right ones. Galen started his medical career in a place that gave him an unusual education, the gladiatorial arena. He was appointed physician to the gladiators of Pergamon when he was in his late 20s, and this was, in a grim way, an extraordinary learning opportunity. Gladiators suffered horrific injuries, deep cuts, crushed bones, penetrating wounds to the chest and abdomen and head. And Galen, who was treating these injuries, got to see things that most physicians of his era never would. He later referred to wounds as windows into the body, and he meant it literally. When a gladiator took a blow to the head and lost the ability to speak, or move a limb, or see, Galen was right there, observing, correlating the location of the injury with the loss of function. He also conducted extensive animal dissections and experiments on pigs, on monkeys, on goats. Roman law at the time generally prohibited the dissection of human cadavers, so Galen worked with animals and extrapolated to humans, which gave him extraordinary insights, but also led to some significant errors that would persist for over 1,000 years. One of his most famous demonstrations involved the laryngeal nerve in a pig. He would expose the nerve during a live dissection, and the pig would be squealing. And then he'd cut the nerve, and the pig would fall instantly silent, even though its mouth was still open, and it was clearly trying to vocalize. The audience would be stunned. Here was visible, undeniable proof that the brain controlled the body through nerves. Cut the nerve, lose the function. It was dramatic, it was convincing, and it was correct. Galen developed a theory of the brain that was remarkably sophisticated for its time, though it was also wrong in important ways. He believed that the key structures were the ventricles, the fluid-filled cavities inside the brain. He thought that a substance he called psychic pneuma, a kind of vital spirit, was produced in the ventricles and then distributed through the nerves to the rest of the body, animating it, giving it sensation and movement. The brain tissue itself, the actual gray and white matter, he considered less important. It was the ventricles and the fluid that mattered. This is called the ventricular theory of brain function. And here's what's wild about it. It dominated thinking about the brain for over 1,000 years. Through the fall of Rome, through the rise of Islamic medicine, through the European Middle Ages, through the Renaissance, physician after physician, scholar after scholar, accepted Galen's basic framework. The ventricles were where the action was. The first ventricle handled sensation and imagination. The middle ventricle handled reason and thought. The rear ventricle handled memory. It was a neat, tidy system. It was also almost entirely wrong, but it was so elegant and so authoritative that nobody seriously challenged it for an extraordinarily long time. And that's something worth sitting with for a moment, because it happens over and over in the history of understanding the brain. Someone brilliant comes up with a framework that explains some of the observations, and then that framework becomes so entrenched that it actually prevents people from seeing what's right in front of them. The brain, it turns out, is so complex that every generation's best theory ends up being, at best, a useful approximation, a map that captures some features of the territory but misses enormous amounts. The Islamic Golden Age, roughly the 8th through the 14th centuries, preserved and expanded on Galen's work in important ways. Scholars like Ibn Sina, known in the West as Avicenna, wrote encyclopedic medical texts that refined the ventricular theory and added clinical observations. Ibn Sina's Canon of Medicine, completed around 1025 CE, was used as a medical textbook in European universities for over 500 years. 500 years. That's an almost incomprehensible span of influence. And while Ibn Sina didn't overturn the ventricular model, he added nuance. He wrote about the relationship between brain injury and specific functional losses, about the effects of drugs on mental states, about the connection between emotional disturbance and physical illness. He was careful, systematic, and deeply thoughtful. But the real revolution in understanding the brain, the moment when people started to actually look at the brain itself, rather than theorizing about invisible spirits flowing through its cavities, didn't come until the Renaissance, when a young Belgian anatomist decided that the only way to understand the human body was to open it up and look. His name was Andreas Vesalius, and he was born in Brussels in 1514 into a family of physicians and pharmacists. Vesalius was, by all accounts, obsessed with anatomy from a young age. There are stories, and some of these might be embellished, but they capture his character, of the young Vesalius collecting animal bones, dissecting mice and stray cats, driven by an almost compulsive need to see the structures inside living things with his own eyes, rather than taking anyone's word for it. And that instinct, that insistence on direct observation over received authority, would change everything. When Vesalius arrived at the University of Paris to study medicine, anatomy was taught in a way that seems almost absurd to us now. The professor would sit in an elevated chair, reading aloud from Galen's texts, while a barber surgeon down below did the actual cutting on a cadaver. The professor rarely even looked at the body. The assumption was that Galen had already described everything correctly, so the dissection was just a formality, a demonstration of what the text said, not an investigation of what was actually there. If the body on the table didn't match Galen's description, the assumption was that the body was anomalous, not that Galen was wrong. Vesalius found this intolerable. He started doing his own dissections, pushing the barber surgeons aside, getting his hands into the cadavers himself, and almost immediately, he started finding discrepancies. Structures Galen described that weren't there. Structures that were there that Galen hadn't mentioned. Relationships between organs that were different from what the texts claimed. And slowly, methodically, Vesalius began to realize something that was genuinely dangerous to say out loud in 16th century Europe. Galen had been wrong. Not about everything, but about a lot of things, and the reason was simple. Galen had mostly dissected animals, not humans, and had assumed the anatomy was the same. It wasn't. In 1543, when he was just 28 years old, Vesalius published De Humani Corporis Fabrica, on the fabric of the human body. And it was a bombshell. 7 volumes of meticulous anatomical description, accompanied by stunning illustrations that were probably produced in the workshop of Titian, though the exact artist is debated. And in those volumes, Vesalius took on the ventricular theory directly. He pointed out that the ventricles of the human brain didn't look particularly special. Animal brains had ventricles too. Similar ventricles. In fact, if the ventricles were the seat of reason and higher thought, why did a cow have them? He didn't have a better theory to offer. Not exactly. But he planted a seed of doubt that would eventually grow into something much bigger. Because once you start questioning the container, you have to start asking what the real machinery is. And that question, what is the brain actually made of, and how does it actually work, would take centuries to answer. But the journey toward that answer? It starts right here, in the decades after Vesalius cracked open the old model. Now, Vesalius himself didn't stick around to finish the revolution he started. His career took a strange turn after the Fabrica was published. The backlash from Galenists was fierce. Some of his former teachers publicly attacked him, accused him of arrogance, of disrespecting the ancients. Vesalius, apparently hurt and frustrated, reportedly burned some of his unpublished manuscripts and took a position as court physician to Emperor Charles V, essentially stepping away from anatomical research. He died in 1564 during a pilgrimage to the Holy Land, under circumstances that are still a little murky. He was only 49, but the seed he planted, that doubt about the ventricles, it didn't die with him. Other anatomists picked up the thread, and over the next century and a half, the focus of brain research shifted in a really important way. Instead of asking what the ventricles do, people started looking more carefully at the brain tissue itself, the actual substance of the brain, the gray stuff, the white stuff, the folds and ridges. And this is where things start to get genuinely interesting, because the brain, when you really look at it, is one of the strangest looking organs in the entire body. Think about it for a second. If you've ever seen a brain, even just a picture, you know it doesn't look like any other organ. Your liver is smooth and dense. Your heart is muscular and compact. Your lungs are spongy and uniform. But the brain? It's wrinkled, deeply, elaborately wrinkled, with these folds called gyri and grooves called sulci that give it that walnut-like appearance. And inside, when you slice it open, you find this stark contrast between the outer layer, which is grayish, and we call it gray matter, and the inner bulk, which is paler, almost white, and we call it white matter. Why? What's the difference? What are those folds for? These are questions that the anatomists of the 1600s were just beginning to ask, and the answers they came up with would lay the groundwork for everything we know about the brain today. One of the key figures in this next phase was an Englishman named Thomas Willis, and Willis is one of those people who deserves to be far more famous than he is. Born in 1621, he lived through the English Civil War, actually served as a royalist soldier for a while, and eventually became a physician and professor at Oxford. Willis was obsessed with the brain, not in a casual way, but in a let's dissect every brain I can get my hands on and map every single structure kind of way. And in 1664, he published a book called Cerebri Anatomae, Anatomy of the Brain, which is arguably the single most important book about the brain written in the 17th century. Maybe ever, up to that point. Here's what Willis did that was different. He didn't just describe the ventricles and move on. He meticulously mapped the structures of the brain itself, the cerebrum, the cerebellum, the brainstem, the network of arteries at the base of the brain that still bears his name, the Circle of Willis. You might have heard that term if you've ever had a medical scan or studied a little anatomy. It's a ring of arteries that connects the major blood supplies to the brain, creating redundancy so that if one artery gets blocked, blood can still reach the tissue through alternate routes. Willis described this beautifully, and he understood, at least intuitively, that the brain needed an enormous blood supply to function. That alone was a clue. Whatever the brain was doing, it was metabolically expensive. But Willis went further than just mapping blood vessels. He made a conceptual leap that was genuinely ahead of his time. He proposed that different parts of the brain were responsible for different functions. This idea, which we now call cerebral localization, seems obvious to us today, but in the 1660s, it was radical. The prevailing view was still that the brain worked as a kind of undifferentiated whole, or that the important stuff happened in the ventricles. Willis said no. He argued that the cerebrum, the big wrinkled upper part, was the seat of thought, memory, and will. The cerebellum, the smaller, densely folded structure at the back and bottom, controlled involuntary movements. The brainstem handled vital functions like breathing and heartbeat. He wasn't right about every detail, but the basic framework? It's remarkably close to what we understand now, more than 350 years later. And here's a detail I love about Willis. He didn't work alone. His dissections were done in collaboration with a brilliant physician named Richard Lower, and, this is the really wonderful part, his illustrations were drawn by Christopher Wren. Yes, that Christopher Wren, the architect who designed St. Paul's Cathedral in London. Before Wren became the most famous architect in England, he was a young scientist at Oxford, and one of his jobs was making detailed, accurate drawings of Willis's brain dissections. So the next time you see a picture of St. Paul's Cathedral, just think, the same hands that designed that dome also drew some of the first accurate maps of the human brain. There's something quietly beautiful about that. Now, Willis's work opened a door, but walking through it would take a long time. Because here's the problem that haunted brain science for the next 2 centuries. You could see that the brain had structure, that it had distinct regions and folds and layers, but you couldn't see what those structures were made of at the microscopic level. The brain, to the naked eye, looks like a fairly uniform mass. Gray matter looks like gray matter. White matter looks like white matter. You can see the big landmarks, the folds, the ventricles, the major divisions. But the fine structure, the actual cellular architecture, that was invisible. And without knowing what the brain was made of at the smallest scale, you couldn't really understand how it worked. This is where we need to talk about the microscope and about a problem that plagued brain research for an absurdly long time. See, the microscope had been around since the late 1500s. By the 1670s, people like Antony van Leeuwenhoek were using microscopes to see bacteria, blood cells, all sorts of tiny structures. You'd think someone would have pointed a microscope at brain tissue early on and figured out what was going on. But brain tissue is uniquely difficult to study under a microscope. Here's why. Most of the brain is water, about 80%. When you slice it thin enough to see through, it's almost transparent. Everything looks the same. The cells blend into each other. The fine structures, the fibers, the connections, they're so thin and so tangled that without some way to make them visible, you're basically looking at a pale, featureless smear. This problem wouldn't be solved until the 1800s, when staining techniques were developed. Chemical dyes that would selectively color certain parts of brain tissue, making individual structures pop out against the background. And that breakthrough is one of the great stories in the history of neuroscience. But we'll get there. For now, just hold that thought. For roughly 200 years after Willis, scientists knew the brain had complex internal structure, but they couldn't see it clearly enough to understand it. So what happened in those 200 years? Well, people got creative. If you can't see the microscopic structure, you can still learn a lot from what happens when things go wrong. And this is where the study of brain injuries and brain diseases becomes the primary engine of discovery. Remember that ancient Egyptian case, the one from the Edwin Smith papyrus, where someone noticed that a wound on one side of the head caused problems on the opposite side of the body? That same basic method, observing what breaks when the brain is damaged, would drive neuroscience forward for centuries. One of the most dramatic examples comes from the late 1700s and into the 1800s, and it involves a really contentious figure named Franz Joseph Gall. Gall was a Viennese physician born in 1758, and he had an idea that was partly brilliant and partly completely wrong, which is kind of the most dangerous combination in science. His brilliant insight was this: different mental functions are localized in different parts of the brain. Sound familiar? It's essentially what Willis had proposed, but Gall took it much further. He believed that the brain was made up of dozens of distinct organs, each responsible for a specific mental faculty. Things like language, mathematical ability, musical talent, courage, even traits like secretiveness or the tendency toward theft. And here's where it went off the rails. Gall believed that when a particular brain organ was well developed, it would be physically larger, and that this enlargement would push outward on the skull, creating a detectable bump. Conversely, an underdeveloped faculty would leave a depression. So by carefully feeling the bumps and contours of a person's skull, you could map their personality, their talents, their moral character. This was phrenology, and for a while, it was enormously popular. Phrenological societies sprang up across Europe and America. People had their heads read the way they might get their palms read today. Employers used it to screen workers. It was taken seriously by some genuinely intelligent people. But it was also, in its specifics, almost entirely wrong. The shape of your skull doesn't reliably correspond to the shape of the brain underneath. Mental faculties don't map neatly onto skull bumps. The whole system was built on anecdotal observations and confirmation bias. But here's the thing about Gall that's really important, and that sometimes gets lost in the justified ridicule of phrenology. His core premise, that the brain is not a uniform organ, that different regions handle different tasks, was correct. And by making that claim loudly and publicly, by insisting on it in the face of people who still thought the brain was an undifferentiated whole, he forced the scientific community to engage with the question of localization. He made it a central debate. And that debate would be settled not by feeling skulls, but by studying what happened to people when specific parts of their brains were damaged. Which brings us to a quiet hospital in Paris in the year 1861, and a patient who could understand everything said to him, but could produce only a single syllable. His name was Louis Victor Leborgne, but everyone at the hospital called him by the only sound he could make: tan. That was it. That was the only syllable he could produce. Tan. Over and over. You could ask him a question, and you could see in his eyes that he understood you perfectly. He'd nod. He'd shake his head. He'd gesture. His comprehension was intact. But when he tried to speak, when he tried to form words and push them out into the world, all that came was that single, repeated sound. Tan. Tan, tan. He'd been at the Bicêtre Hospital in Paris for over 20 years by the time the right doctor finally paid attention to him. And during those decades, he'd gradually lost more and more function. First, the speech, then movement on his right side. Then his vision began to deteriorate. By the time he became truly relevant to the history of neuroscience, Le Borgne was bedridden, suffering from gangrene and very near death. The doctor who changed everything was Paul Broca. He was a surgeon, an anatomist, a polymath, really. The kind of person who seemed to be interested in everything and good at most of it. Broca had been following the localization debate closely. There was a fierce argument raging at the time within the Anthropological Society of Paris, which Broca himself had founded just a couple of years earlier, about whether specific mental functions could really be pinned to specific brain regions. One camp, led by a man named Pierre Gratiolet, argued that the brain worked as a whole, that you couldn't carve it up into functional zones. The other camp, which included Broca's colleague Ernest Aubertin, insisted that speech, at least, was localized in the frontal lobes. Aubertin had even made a dramatic public promise. If anyone could show him a case where the frontal lobes were destroyed, but speech was preserved, he'd abandon his position entirely. So when Le Borgne's condition worsened in April of 1861, and it became clear he didn't have long to live, Broca saw an opportunity. He examined the patient carefully, documented his symptoms, and then, when Le Borgne died on April 17th, Broca performed an autopsy. And what he found in that brain was extraordinary. Or rather, what he didn't find. There was a lesion, a region of significant damage in the left frontal lobe. Specifically, it was in the posterior portion of the third frontal convolution, a particular fold of cortex on the left side toward the front of the brain. The tissue there was softened, destroyed, and Broca argued that this was the seat of articulate speech. Not of language comprehension, mind you. Le Borgne had understood language just fine. But the ability to produce speech, to coordinate the complex muscular movements needed to form words, that was what had been destroyed. Now, you might think one case wouldn't be enough to convince anyone, and you'd be right to be skeptical. But Broca didn't stop there. Within the next few years, he documented several more patients with similar profiles, people who could understand speech but couldn't produce it, and who, upon autopsy, showed damage in that same region of the left frontal lobe. Case after case, the lesion was in the same place. And crucially, it was almost always on the left side. That detail is huge. It meant not only that speech production was localized to a specific brain region, but that the two hemispheres of the brain weren't mirror images of each other in terms of function. The left hemisphere, at least for most people, was dominant for language. This was one of the first clear demonstrations of what we now call lateralization, the idea that the two halves of your brain, which look so similar from the outside, actually specialize in different things. That region Broca identified? We still call it Broca's area today. And the condition, where someone can understand language but can't produce fluent speech, is called Broca's aphasia. If you've ever known someone who suffered a stroke affecting the left side of their brain and afterward struggled to get words out, speaking in short, effortful, telegraphic phrases while clearly understanding what was said to them, that's Broca's aphasia. it's still one of the most common consequences of left hemisphere strokes. But here's what's really fascinating about the timing. Just about 13 years after Broca's discovery, in 1874, a German physician named Karl Wernicke, he was only 26 years old at the time, which is remarkable, published a paper describing essentially the mirror image of Broca's patients. Wernicke had found patients who could speak fluently. Their words came out smoothly, with normal rhythm and intonation. Grammatically, their sentences sounded fine, but the words themselves were often wrong, sometimes nonsensical. They'd substitute one word for another, or use words that didn't exist. And critically, they couldn't understand spoken language. You'd talk to them, and they'd look at you blankly, or respond with a stream of fluent but meaningless speech. It was the opposite pattern from Le Borgne. And when Wernicke examined the brains of these patients, the damage was in a different spot, farther back, in the upper part of the left temporal lobe, near where it meets the parietal lobe. So now you had two distinct language centers. Broca's area in the left frontal lobe for producing speech, Wernicke's area in the left temporal lobe for comprehending speech. And Wernicke, being a sharp thinker, didn't just describe the two areas in isolation. He proposed a model, a kind of circuit. He suggested that the two regions were connected by a bundle of nerve fibers, and that language processing involved information flowing between them. You hear words, and Wernicke's area processes the meaning. When you want to speak, the information travels forward along that fiber bundle to Broca's area, which organizes the motor commands to produce the words. And Wernicke predicted, this is the beautiful part. He predicted that if you damaged not the two areas themselves, but the connection between them, you'd get a third kind of aphasia. The patient would be able to understand speech and would be able to produce fluent speech, but they wouldn't be able to repeat things back to you, because the pathway from comprehension to production would be severed. And this condition, called conduction aphasia, was indeed later confirmed. It exists. Wernicke predicted it from theory before it was observed clinically. Think about what that means for a second. By the 1870s, barely a decade after Broca's first report on Le Borgne, scientists had gone from debating whether the brain had specialized regions at all to mapping specific language circuits and predicting new disorders based on the wiring diagram. That's an incredible leap. And it all came from the same basic method, studying what goes wrong when specific parts of the brain are damaged. The lesion method, as it's sometimes called. You can't ethically damage a healthy person's brain to see what happens, but nature and disease and accidents do it for you. And if you're careful and systematic about documenting the symptoms and then examining the brain after death, you can start to build a map. And that map kept growing. Around the same time, Broca and Wernicke were working out language circuits. Other researchers were tackling movement. In the late 1860s and early 1870s, two German physicians, Gustav Fritsch and Eduard Hitzig, did something that had never been done before, or at least never done carefully and systematically. They electrically stimulated the exposed brain of a living dog and showed that stimulating specific spots on the cortex produced specific movements. Touch one spot, and the dog's left foreleg would twitch. Touch another, and its jaw would move. This was direct, experimental proof that the cortex controlled movement, and that it did so in an organized, mapped-out fashion. Now, the story of how they did this experiment is one of those details that tells you a lot about the state of science at the time. Fritsch and Hitzig didn't have a proper laboratory. There's a widely repeated account. And some historians debate the exact details, but the spirit of it seems right. That they conducted at least some of their early experiments on a dressing table in Fritsch's home. A dressing table. They were mapping the motor cortex of a living animal on bedroom furniture. The resources available to researchers in that era were sometimes astonishingly modest compared to what we'd consider necessary today. But their results were undeniable, and they opened up a whole new approach. If you could stimulate the brain electrically and produce specific effects, then you had a tool that went beyond just waiting for nature to damage someone's brain. You could probe the brain actively. And over the following decades, researchers refined this approach extensively. The most famous figure in this next phase was probably David Ferrier, a Scottish neurologist working in London, who in the 1870s conducted extensive electrical stimulation experiments on animal brains, primarily monkeys, and produced detailed maps of which cortical regions controlled which body parts. Ferrier's work was controversial, partly because of growing public concern about animal experimentation, and he was actually prosecuted under the Cruelty to Animals Act of 1876, though he was ultimately acquitted because the experiments had technically been performed by a colleague. But the maps held up, and they revealed something beautiful and strange about how the brain organizes movement. The motor cortex, that strip of brain tissue running roughly from ear to ear across the top of your head, contains a kind of map of your entire body. But it's not a proportional map. It's distorted. The parts of your body that require the finest, most precise motor control, your hands, your fingers, your lips, your tongue, take up a disproportionately large amount of cortical real estate. Your entire trunk, by contrast, which is massive but doesn't need the same fine-grained control, gets a relatively small patch. If you were to draw a human figure where each body part was sized according to how much motor cortex it occupies, you'd get this bizarre, wonderful creature with enormous hands, a huge face with gigantic lips, and a tiny little torso. This distorted figure has a name, the motor homunculus. Homunculus just means little man in Latin, and there's a corresponding sensory homunculus for the somatosensory cortex, right next door, which maps out touch and sensation with the same kind of distortion. Your fingertips and your lips, which are exquisitely sensitive, dominate the map. These homunculi, and I love that the plural of homunculus is homunculi, it just sounds right, were most famously mapped in detail by a Canadian neurosurgeon named Wilder Penfield in the 1930s, 40s, and 50s. And Penfield's story is one of those cases where a medical necessity opened a window into fundamental science in a way nobody could have planned. Penfield was a neurosurgeon treating patients with severe epilepsy, the kind that didn't respond to medication. For these patients, surgery was sometimes the only option. You go in and remove the small region of brain tissue where the seizures originate. But to do that safely, you need to know exactly what that region does, because you don't want to remove the part of the cortex that controls your patient's right hand or their ability to speak. So, Penfield developed a technique where he would operate on patients who were awake, The brain itself has no pain receptors. So, once you get through the skull and the membranes, the patient doesn't feel the probe touching their cortex, and he would systematically stimulate different spots with a tiny electrode, asking the patient what they experienced. Touch this spot. I feel a tingling in my left thumb. Touch that spot. My right foot just moved. Touch another spot, and the patient might suddenly hear music or smell something or experience a vivid memory from childhood. Penfield was mapping the living human brain in real time with the patient as his guide, and the maps he produced, those homunculi, became some of the most iconic images in all of neuroscience. But Penfield's work revealed something beyond just motor and sensory maps. Those moments when a patient would suddenly relive a memory, or hear a fragment of a song, or feel a wash of emotion, those hinted at something far more complex and mysterious about what the cortex was doing. It wasn't just a switchboard for inputs and outputs. It was storing experiences. It was generating the felt texture of a life and understanding how it did that, how billions of individual cells could somehow give rise to memory, to emotion, to consciousness itself, that required getting down to a level of detail that Broca and Wernicke and even Penfield couldn't reach. It required understanding the basic building block of the brain, the individual nerve cell. And the story of how we came to understand that cell is one of the great scientific rivalries in history, a fight between two brilliant men who looked at the same tissue under the same kind of microscope and saw fundamentally different things. One of them was an Italian, the other was Spanish, and the argument between them would reshape everything we thought we knew about the brain's fundamental architecture. And to understand why their disagreement mattered so much, you need to appreciate just how frustrating it was to study brain tissue under a microscope before the 1870s. Because here's the problem. Brain tissue, when you slice it thin enough to put on a glass slide, is essentially transparent. It's a pale, featureless mush. You're looking at the most complex structure in the known universe, and under a basic microscope it looks like nothing, just a wash of gray. You can't see individual cells. You can't see connections. You can't see structure. It's like trying to read a book printed in white ink on white paper. This is why the microscopic study of the brain lagged so far behind other organs. By the mid-1800s, anatomists had beautifully detailed maps of muscle fibers, liver cells, kidney tubules, tissues that took up stains well and had clear, visible structure. But the brain resisted. It kept its secrets. And the person who accidentally cracked this problem was Camillo Golgi, a young Italian physician working in the most unlikely of laboratories. Golgi was born in 1843 in a small town in Lombardy, the son of a doctor. He studied medicine at the University of Pavia, and after graduating, he took a position at a chronic disease hospital, essentially an asylum, in the town of Abbiategrasso, about 20 miles southwest of Milan. This was not a prestigious research appointment. This was a poorly funded institution for long-term patients, and Golgi was there as a clinician, not a scientist. But he was obsessed with the structure of the nervous system, and he set up a makeshift laboratory in, and this detail always gets me, in the kitchen of his apartment. He was doing histology, the study of tissues under the microscope, in his kitchen, at night, by candlelight. Think about that. some of the most important discoveries in the history of neuroscience came out of a kitchen in a provincial Italian hospital. And sometime around 1873, Golgi stumbled onto something extraordinary. He was experimenting with different chemical treatments for brain tissue, trying to find something, anything, that would make the cells visible. And he discovered that if you hardened brain tissue in potassium dichromate and then soaked it in silver nitrate, something magical happened. The silver compound would precipitate inside certain nerve cells, staining them a deep, inky black against a clear yellow background. And here's the truly remarkable part. For reasons that are still not fully understood to this day, this reaction, which became known as the black reaction, or la reazione nera, only stains a tiny fraction of the cells in any given slice, maybe 1 or 2%. If it stained all of them, you just see a solid black smear because the cells are packed so densely. But because it randomly selects just a few, you can suddenly see individual nerve cells in their entirety, standing out in sharp silhouette against everything around them. Their cell bodies, their long branching extensions, all of it rendered in exquisite detail for the first time in human history. It was like someone had turned on a light in a dark room. Golgi could now see what no one had ever seen before, the actual shape of a nerve cell. And what he saw was astonishing. These weren't simple round blobs like blood cells or neat bricks like skin cells. They were wildly elaborate structures with long, thin fibers extending outward in every direction, branching again and again, reaching across what seemed like enormous distances in the tissue. The complexity was breathtaking. But here's where Golgi's interpretation comes in, and this is crucial. When Golgi looked at those beautiful stained preparations, he saw a network. He saw fibers from different cells reaching out and apparently merging into one another, forming what he described as a continuous web, a reticulum, a diffuse net of interconnected fibers through which signals could flow in any direction, like water through a sponge. In Golgi's view, the brain was not made up of discrete individual units. It was one giant fused network. The fibers of one cell blended seamlessly into the fibers of the next. This became known as the reticular theory, and it was a perfectly reasonable interpretation of what Golgi was seeing. Under his microscope, those fibers really did look like they were touching, merging, forming a continuous mesh. Now enter Santiago Ramon y Cajal. And if Golgi's story is compelling, Cajal's is extraordinary. Born in 1852 in the tiny village of Petilla de Aragon in the Spanish Pyrenees, Cajal was, by his own account, a terrible student and a constant troublemaker as a child. His father was a surgeon, a self-made man who had clawed his way up from poverty, and he wanted Santiago to study medicine. Young Cajal wanted to be an artist. He was obsessed with drawing, with capturing the world in precise, detailed images. His father considered this a waste of time, and, in one of those parenting decisions that sounds almost comically harsh, actually had the boy apprenticed to a shoemaker and then a barber to try to beat the art out of him. It didn't work, but Cajal eventually did study medicine, partly to please his father, and here's the beautiful irony. His artistic talent, that thing his father tried to suppress, turned out to be the very skill that would make him one of the greatest scientists who ever lived. Cajal didn't encounter Golgi's staining method until 1887, about 14 years after Golgi had published it. A colleague showed him a preparation made with the black reaction, and Cajal was immediately captivated. He went home and began using the technique obsessively, applying it to every type of nervous tissue he could get his hands on. But Cajal did something clever that Golgi hadn't done systematically. He applied the stain to embryonic and very young animal brains, not just adult tissue. And this made all the difference. In a developing brain, nerve cells are fewer in number, and their branching extensions are simpler, less tangled. It's like the difference between trying to trace individual vines in a mature, overgrown jungle versus looking at seedlings in a nursery. In the younger tissue, you could see individual cells much more clearly. You could follow their fibers from beginning to end. And what Cajal saw was fundamentally different from what Golgi had concluded. Where Golgi saw fusion and continuity, Cajal saw separation. He saw that the fibers of one nerve cell came very, very close to the fibers of the next, tantalizingly close, but they did not actually merge. There was a gap. Each nerve cell was its own individual entity, a discrete unit with its own cell body, its own branching inputs, its own long output fiber, and it communicated with other cells not by direct physical fusion, but by transmitting signals across that tiny gap. The nerve cell was an independent unit. The brain was not a continuous web. It was a society of billions of individual cells, each one separate, each one making specific connections with specific neighbors. This was the neuron doctrine, and it was revolutionary. Cajal drew what he saw with the hand of a master artist. His illustrations of nerve cells are still used in textbooks today, over a century later, because they are that accurate and that beautiful. He drew with a precision and clarity that photographs of the era couldn't match. Every branching dendrite, every swelling along an axon, every point of near contact between cells, he captured it all. His drawings weren't just scientific records. They were acts of seeing, of understanding made visible on paper. Now, you might think this would have been settled quickly. Cajal had better evidence, cleaner preparations, a more elegant interpretation. But Golgi did not agree. Not then, not ever. And this is where the rivalry becomes genuinely dramatic. In 1906, the Nobel Prize in Physiology, or Medicine, was awarded jointly to both men, Golgi and Cajal, together, for their work on the structure of the nervous system. They traveled to Stockholm. They gave their Nobel lectures. And Golgi used his lecture, his Nobel lecture, the most prestigious scientific address he would ever give, to attack Cajal's neuron doctrine and defend the reticular theory. He stood up in front of the assembled scientific world and essentially said, Cajal is wrong. The brain is a continuous network, not a collection of individual cells. Cajal was reportedly furious, and history has shown that Cajal was right. The neuron doctrine, the idea that the brain is composed of discrete individual cells, which we now call neurons, that communicate across gaps we now call synapses, became one of the foundational principles of all modern neuroscience. Every single thing we understand about how the brain processes information, how memories form, how diseases like Alzheimer's and Parkinson's work, how psychiatric medications function, all of it rests on the fact that the brain is made of individual neurons that talk to each other across synaptic gaps. Golgi invented the tool that made the discovery possible, but but he couldn't see what his own tool was showing him. Kajal could. And the difference was partly technique, those embryonic preparations, and partly, honestly, artistic vision. Kajal could see because he knew how to look. But here's the thing that's easy to miss in the drama of the rivalry. Kajal's neuron doctrine immediately raised a question that was, in some ways, even harder than the one it answered. If neurons are separate, if there's a physical gap between them, then how does a signal get from one neuron to the next? How does information cross that gap? Because the electrical nature of nerve signals was already becoming understood by this point. People knew that nerves carried some kind of electrical impulse. Luigi Galvani had shown that back in the 1780s with his famous frog leg experiments, and by the late 1800s, scientists had a reasonable understanding that nerve fibers conducted electrical signals along their length, like a wire. But a wire has to be connected to something. If Cajal was right, and neurons weren't physically fused, then the signal had to somehow jump across empty space. How? This question consumed neuroscience for the next several decades, and it produced another rivalry. Not quite as personal as Golgi versus Kajal, but scientifically just as consequential. On one side were the people who believed the signal crossed the gap electrically, that the electrical impulse in one neuron somehow sparked an electrical impulse in the next, like a spark jumping between two wires held close together. On the other side were the people who believed the signal crossed chemically, that the first neuron released some kind of chemical substance into the gap, and that chemical substance triggered the next neuron to fire. The electrical camp versus the chemical camp. And the person who would tip the scales was a pharmacologist in Austria who claimed the answer came to him in a dream, literally in his sleep, which feels almost too perfect given where you are right now. His name was Otto Lowy, and the experiment he designed was so elegant, so beautifully simple, that it almost doesn't seem like it should have settled anything. But it did. So here's the story. It's 1921, and Otto Lowy is a professor of pharmacology at the University of Graz in Austria. He's been thinking about this problem of synaptic transmission for years, not casually, but deeply, the way it sits in the back of your mind and won't let go. He already suspected that chemical substances were involved. In fact, the idea wasn't entirely new. Back in 1904, a British physiologist named Thomas Renton Elliot had suggested that adrenaline might be released by certain nerves to stimulate muscles and organs. And Henry Hallett Dale, another British scientist, had been doing important work on a substance called acetylcholine, showing that it could mimic the effects of stimulating certain nerves. So the clues were there. But nobody had proven it. Nobody had shown in a clean experiment that a nerve actually releases a chemical that then acts on another cell. It was still just a hypothesis, and the electrical camp had their own arguments. Mainly, that chemical transmission would be too slow. Nerve signals can cross from one neuron to the next in less than a millisecond. How could a chemical process possibly work that fast? Lowy knew he needed a decisive experiment. And according to his own account, and he told this story many times, so we have it from multiple sources, the idea for that experiment came to him in the middle of the night. He woke up from a dream, grabbed a piece of paper, scribbled down notes, and went back to sleep. When he woke up in the morning, he couldn't read his own handwriting, couldn't remember the dream. He was devastated. But the next night, the dream came back. And this time, he didn't bother writing it down. He got up, went straight to his laboratory at 3 a.m, and performed the experiment right then and there. Here's what he did. He took 2 frog hearts. Both were still beating, as frog hearts will do for a while after they're removed if you keep them in the right solution. The first heart still had its vagus nerve attached. That's the nerve that, when stimulated, slows the heart down. The second heart had no nerve attached at all, just sitting there beating on its own. Lowy stimulated the vagus nerve on the first heart, and sure enough, it slowed down. That was expected. But then he took the fluid, the saline solution that the first heart had been sitting in, and transferred it to the second heart. The second heart slowed down too. Think about what that means. Nothing touched the second heart except liquid. No electrical connection, no wire, no nerve. Just fluid that had been bathing a heart whose nerve had been stimulated. Something had been released into that fluid, some chemical substance, and that substance alone was enough to slow the second heart. Lowy called it Vagusstoff, which just means vagus substance. It was later identified as acetylcholine, the same compound Henry Dale had been studying. That experiment, performed in the middle of the night by a man chasing a dream he'd already lost once, is one of the most important in the history of neuroscience. It demonstrated clearly and undeniably that nerves communicate using chemical messengers. Lowy and Dale would share the Nobel Prize in Physiology or Medicine in 1936 for their work on chemical transmission of nerve impulses. And the concept they established, that neurons talk to each other by releasing chemicals across the gap between them, became one of the foundational principles of how we understand the brain. Now, that gap has a name. It's called the synapse, a term coined by the British physiologist Charles Sherrington back around 1897, before anyone had even proven how signals crossed it. Sherrington was studying reflexes, those automatic responses, like pulling your hand away from something hot. And he realized that reflexes involved chains of neurons working together. He needed a word for the junction between them, so he borrowed from the Greek word synapsis, meaning to clasp together, the synapse. And the chemicals that cross it? Those are neurotransmitters. Acetylcholine was the first one identified, but it was far from the last. And here's where things start to get really interesting for your everyday life. Because once scientists understood that neurons communicate chemically, a whole new world opened up. If the brain runs on chemicals, then you can change how the brain works by changing those chemicals. That's the entire basis of psychopharmacology, of every psychiatric medication, every antidepressant, every anti-anxiety drug, every treatment for Parkinson's disease or schizophrenia. All of it traces back to this insight that neurons release specific chemicals, and those chemicals can be mimicked, blocked, enhanced, or depleted. Let's follow that thread for a moment, because the story of how we discovered the brain's chemical messengers is one of the great detective stories of 20th century science. After acetylcholine, the next major neurotransmitter to be understood was norepinephrine, closely related to adrenaline, the substance Eliot had speculated about decades earlier. Then, In the 1950s, a Swedish scientist named Arvid Carlsson made a discovery that would eventually earn him a Nobel Prize and transform the treatment of one of the most devastating neurological diseases in the world. Carlsson was studying a chemical called dopamine. At the time, most scientists thought dopamine was just a precursor, a building block that the body used to make norepinephrine, but not important on its own. Carlsson disagreed. He had been experimenting with a drug called reserpine, which was known to deplete certain chemicals from the brain and cause animals to become rigid and immobile, symptoms that looked a lot like Parkinson's disease. When Carlsson gave these depleted animals a precursor to dopamine called L-Dopa, they recovered. Their movement came back. And when he measured dopamine levels in the brain, he found that it was concentrated in a specific region called the striatum, a part of the brain deeply involved in controlling movement. The implication was enormous. Parkinson's disease, with its tremors and rigidity and progressive loss of motor control, might be caused by a loss of dopamine in the brain. And if that was true, then maybe you could treat it by giving patients L-Dopa to replenish what they'd lost. That's exactly what happened. In the 1960s, clinical trials of L-Dopa in Parkinson's patients showed dramatic improvements. People who had been frozen, unable to move, were suddenly walking again. It wasn't a cure. The disease still progressed, but it was the first truly effective treatment, and it remains a cornerstone of Parkinson's therapy to this day. So here you have this beautiful chain of understanding. Kajal shows that neurons are separate cells. Lowy proves they communicate chemically. Scientists identify specific neurotransmitters. And then clinicians figure out that diseases can result from imbalances in those chemicals, and that you can treat those diseases by correcting the imbalance. Each insight builds on the last, like layers of brick. But the neurotransmitter story also reveals something humbling about how the brain works, which is that it's not just one chemical doing one thing. The brain uses dozens of different neurotransmitters, and each one can have different effects depending on where it's released and what kind of receptor it binds to. Dopamine doesn't just control movement. It's also deeply involved in motivation, reward, pleasure, and learning. That's why drugs that flood the brain with dopamine, like cocaine and amphetamines, produce intense euphoria, but can also be devastatingly addictive. Too little dopamine in the motor system gives you Parkinson's. Too much dopamine activity in certain other brain circuits is thought to contribute to the hallucinations and delusions of schizophrenia. Same molecule, completely different effects depending on where in the brain you're talking about. Serotonin is another one. You've probably heard of it. It's often called the feel-good chemical, which is a massive oversimplification, but not entirely wrong. Serotonin is involved in mood regulation, sleep, appetite, and a dozen other things. Most modern antidepressants, the SSR is, like fluoxetine, which you might know as Prozac, work by preventing serotonin from being reabsorbed after it's released, so it stays in the synapse longer and has a stronger effect. The first SSRI was introduced in the late 1980s, and it genuinely changed millions of lives. But it also highlighted how much we still don't understand, because SSR is increased serotonin levels within hours, Yet the therapeutic effects on mood typically take weeks to appear. If depression were simply not enough serotonin, you'd expect the drugs to work immediately. The fact that they don't tells us the real story is more complicated, that the brain is adapting, rewiring, adjusting its sensitivity to the chemical over time. The drug isn't just flipping a switch, it's nudging a system, and the system slowly recalibrates. And that idea, that the brain recalibrates, that it changes in response to experience and input, brings us to one of the most profound discoveries of the 20th century, one that overturned a belief that had dominated neuroscience for decades. For most of the 1900s, the prevailing view was that the adult brain was essentially fixed. You were born with a certain number of neurons. They wired themselves up during childhood, and after that, the structure was set. You could learn new things, sure, but the basic hardware didn't change. If brain tissue was damaged, it was gone forever. This was considered settled science. Santiago Ramon y Cajal himself had written, somewhat poetically, that in the adult brain, the nerve paths are something fixed, ended, immutable. Everything may die, nothing may be regenerated. It's a beautiful sentence, and it turned out to be wrong. Not entirely wrong. Cajal was right that the brain has far less regenerative capacity than, say, the liver or the skin. But the idea that the brain can't change its structure at all? That was overturned by a concept that we now call neuroplasticity, and the story of how we came to understand it involves some surprising characters and some experiments that at first almost nobody believed. One of the earliest hints came from a psychologist named Donald Hebb, working at McGill University in Montreal in the late 1940s. Hebb proposed a theory about how learning might work at the level of individual neurons, and he summarized it with a phrase that became one of the most famous in all of neuroscience, a phrase so catchy that it's almost a slogan. He said that when one neuron repeatedly helps fire another neuron, the connection between them gets stronger. Or as it's usually paraphrased, neurons that fire together, wire together. Hebb published this idea in 1949 in a book called The Organization of Behavior, and at the time, it was purely theoretical. He had no way to prove it. But the idea was powerful because it offered a mechanism for learning and memory that was grounded in the physical structure of the brain. If connections between neurons could get stronger with use, then experience could literally reshape the brain's wiring. And if connections could get stronger, maybe they could also get weaker. Maybe the brain was constantly adjusting itself, strengthening some pathways, pruning others, based on what it encountered in the world. It took decades to confirm Hebb's intuition, but when the evidence finally came, it came from a place you might not expect, the sea. Specifically, from the humble sea slug, a creature called Aplysia californica, and from a man named Eric Kandel, who would spend the better part of his career studying it. Kandel is one of those figures in science whose personal biography is inseparable from the work he did. He was born in Vienna in 1929 into a Jewish family. When the Nazis annexed Austria in 1938, he was 9 years old. His family eventually fled to the United States, settling in Brooklyn, and Kandel later said that the experience of watching a civilized society descend into brutality shaped his entire intellectual life. He wanted to understand the human mind, what drives people, what makes memory so powerful, why certain experiences imprint themselves on us and never let go. He started out interested in psychoanalysis, actually. He was drawn to Freud, which makes a kind of poetic sense, given that Freud himself had started as a neuroanatomist. But Kandel gradually shifted toward a more biological approach. He wanted to find the physical basis of memory, not the metaphor, not the abstraction, the actual cellular mechanism. And here's where the sea slug enters the picture. Kandel made a decision in the early 1960s that a lot of people thought was, frankly, a little crazy. He decided that if he wanted to understand how memory works at the level of individual neurons, he needed to study the simplest possible nervous system he could find. The human brain, with its roughly 86 billion neurons, was just too complex. You couldn't track what was happening at individual synapses in a system that dense. So he went looking for a creature with a nervous system, simple enough that you could actually watch individual cells in action. Aplysia californica has about 20,000 neurons, and some of them are enormous, visible to the naked eye. You can stick an electrode into a single neuron in this animal and record what it does. Now, a sea slug doesn't exactly have what you and I would call memories. It doesn't remember its first day of school, but it does learn. It has a simple reflex. If you touch its siphon, a little tube it uses for breathing, it withdraws its gill. It's like pulling your hand away from a hot stove, just much simpler. And here's what Kandel showed. If you repeatedly touch the siphon without any harmful consequence, the slug gradually stops withdrawing its gill. It habituates. It learns that the touch is harmless. Conversely, if you pair the touch with something unpleasant, a mild shock to the tail, the slug becomes sensitized. It starts withdrawing its gill more vigorously, even to a gentle touch, and it keeps doing this for a while afterward. That's a form of learning. Simple, yes, but learning. What Kandel and his colleagues were able to do painstakingly over years of work through the 1960s and 1970s was trace exactly what was happening at the synapses during these two forms of learning. During habituation, the synaptic connections between the sensory neuron and the motor neuron got weaker. Less neurotransmitter was released each time. During sensitization, those connections got stronger. More neurotransmitter flooded the gap. And here's the part that confirmed Hebb's prediction from decades earlier. The changes weren't just fleeting. If you repeated the training enough times, the synaptic changes became long-lasting. The cell actually grew new synaptic connections. The physical structure of the neuron changed. Think about what that means. A sea slug, through experience, was literally rewiring its own nervous system. New connections were sprouting where none had existed before. And if a sea slug could do it, with its 20,000 neurons, then maybe, probably, the vastly more complex human brain could do something similar, just on a scale that was orders of magnitude more elaborate. Kandel won the Nobel Prize in Physiology or Medicine in 2000 for this work, and it's one of those Nobel Prizes that feels deeply earned. He took Hebb's theoretical idea, neurons that fire together, wire together, and showed the physical reality of it, synapse by synapse, molecule by molecule. But Kandel's sea slug work was only part of the story. Around the same time, in the early 1970s, two researchers in England Timothy Bliss and Terje Lomo discovered something remarkable in the brains of rabbits. They found that if you stimulated a neural pathway with a rapid burst of electrical pulses, the synapses along that pathway became stronger, and they stayed stronger for hours, even days. They called this phenomenon long-term potentiation, or LTP, and it was essentially Hebb's rule playing out in a mammalian brain. Stimulate two connected neurons together repeatedly, and the connection between them gets physically, measurably stronger. The synapse becomes more efficient. It takes less signal to trigger a response. Long-term potentiation turned out to be especially prominent in a brain structure called the hippocampus, and this is where several threads of our story start weaving together in a really satisfying way. The hippocampus, the name comes from the Greek word for seahorse, because someone thought it looked like one, had already been identified as crucial for memory, thanks to one of the most famous and, honestly, most heartbreaking case studies in the history of neuroscience. In 1953, a young man named Henry Molaison, who was known in the scientific literature for decades only as Patient HM, to protect his privacy, underwent brain surgery in Hartford, Connecticut. Henry had suffered from severe epilepsy since childhood, and his seizures were so debilitating that a neurosurgeon named William Beecher Scoville proposed a radical operation, removing the hippocampus and surrounding structures from both sides of Henry's brain. The surgery did reduce his seizures, but it came at a devastating cost. Henry lost the ability to form new long-term memories. He could remember his childhood. He could carry on a conversation. His personality was intact. His intelligence was normal. But the moment you left the room and came back, he had no idea who you were. He would read the same magazine over and over, each time as if for the first time. He could learn new motor skills. If you taught him to trace a shape in a mirror, he'd get better at it over days. But he had no memory of the practice sessions. He'd sit down, perform better than the day before, and have absolutely no conscious recollection of ever having done the task. Henry Molaison lived like this for 55 years, until his death in 2008 at the age of 82. And the scientific community studied him extensively throughout his life, with his consent and the consent of his family. What his case revealed was profound. The hippocampus isn't where long-term memories are stored, exactly, but it's essential for creating them. It's like a gateway. Information comes in through your senses, gets processed in various cortical areas, and then the hippocampus binds it all together into a coherent memory, and over time, helps transfer it to more permanent storage in the cortex. Without the hippocampus, new experiences just wash through you. They never solidify. So when Bliss and Lomo found that long-term potentiation was especially robust in the hippocampus, the implications were electric. Here was a brain structure known to be essential for forming memories, and it was exhibiting exactly the kind of synaptic strengthening that Hebb had predicted would underlie learning. The pieces were fitting together. Now, I should be honest with you. The full picture of how memory works is still not completely understood. We know LTP is important. We know the hippocampus is crucial for consolidation. We know that memories eventually become less dependent on the hippocampus and more distributed across the cortex, a process called systems consolidation that can take weeks, months, even years. And here's something beautiful. This consolidation process appears to happen preferentially during sleep, which means that right now, as you're lying here listening to this, your brain is probably doing some of this work. The experiences of your day are being replayed, sorted, strengthened, or pruned. The hippocampus is talking to the cortex, deciding what's worth keeping. There's actually a lovely symmetry in that. You're listening to a story about how the brain forms memories, and your brain is simultaneously forming memories, or deciding not to, using the very mechanisms we're describing. If you drift off, your sleeping brain might consolidate some fragment of this, some image of Eric Kandel and his sea slugs, some echo of Henry Molaison reading the same magazine for the 10,000th time. Or it might let it go. Your hippocampus will decide. But let's come back to neuroplasticity more broadly, because it's not just about memory. The discovery that the brain can physically rewire itself had implications that went far beyond learning and recall. It changed how we think about recovery from injury, about therapy, about aging, about what it means to practice a skill. One of the most dramatic demonstrations came from a neuroscientist named Michael Merzenich, working at the University of California, San Francisco, starting in the 1980s. Merzenich was studying the somatosensory cortex, that strip of brain tissue that Penfield had mapped decades earlier with his gentle electrical probe, the one that produced the distorted homunculus with its enormous hands and lips. Merzenich wanted to know, is that map fixed? Is the hand area always the hand area? Or can it change? He designed experiments with monkeys in which he either amputated a finger or, more often, trained the monkeys to use specific fingers intensively. For example, having them repeatedly touch a spinning disc with just their middle fingers to get a food reward. Then he mapped their cortex, and what he found was stunning. The cortical area devoted to the trained finger had expanded. It had physically taken over territory that had previously belonged to neighboring fingers. The map had redrawn itself based on use, and the reverse was also true. If a finger was amputated or a nerve was cut, the cortical territory that had served that finger didn't just go dark. It got colonized by neighboring areas. The brain didn't waste real estate. It reallocated. This was neuroplasticity in action, visible and measurable in the adult brain. And it meant that Cajal's famous dictum, that the adult brain is fixed and immutable, was definitively wrong. The brain is not a static organ. It's constantly remodeling itself, responding to experience, to injury, to demand. Every time you practice a musical instrument, every time you learn a new language, every time you navigate an unfamiliar city, your cortical maps are shifting, subtly, incrementally, but really and truly changing their physical structure. Merzenich went on to apply this understanding to practical problems, developing brain training programs, working on cochlear implants, exploring how plasticity might be harnessed to help people recover from strokes or cope with learning disabilities. And his work inspired a whole generation of researchers to ask similar questions. If the sensory cortex can reorganize, what about the motor cortex? What about higher cognitive areas? How far does plasticity go? The answers, it turned out, went further than almost anyone had imagined. In the late 1990s, researchers discovered something that would have been considered heretical just a decade earlier. Something that contradicted not just Cajal's dictum about fixed wiring, but an even more fundamental assumption. They found that the adult brain can grow entirely new neurons. The process is called neurogenesis, and it was first convincingly demonstrated in the hippocampus of adult rats by Elizabeth Gould at Princeton, and, independently, by Fred Gage and his colleagues at the Salk Institute. For most of the 20th century, the dogma had been absolute. You're born with all the neurons you'll ever have, and from there, it's a slow decline. But Gould and Gage showed that the hippocampus, that same structure so critical for memory, was quietly producing new neurons throughout life. The discovery was controversial. Some researchers pushed back hard. The evidence in humans took longer to establish, and even now, there's active debate about how much neurogenesis occurs in the adult human brain and how functionally significant it is. But the principle was established. The brain is not just rewiring existing connections. In at least some regions, it's building new cells from scratch. And this brings us to a question that's been quietly lurking beneath everything we've talked about so far. A question that connects the chemical messengers, the synaptic plasticity, the cortical maps, and the new neurons into something bigger. It's the question of how all of this gives rise to what you're experiencing right now, this very moment, as you lie here in the dark. The question of consciousness. How does a 3-pound organ made of cells and chemicals and electrical impulses produce the subjective experience of being you? How does matter become mind? It's arguably the deepest question in all of science, and the history of how people have tried to answer it is as fascinating as the question itself. Because for most of human history, people didn't even frame it as a problem. If you go all the way back to Descartes, René Descartes, writing in the 1640s, you find someone who at least had the courage to state the puzzle clearly, even if his answer created more confusion than it resolved. Descartes looked at the body and saw a machine. He was actually quite modern in that respect. He understood that muscles contracted, that nerves carried signals, that reflexes could operate automatically. But he couldn't accept that the mind, the thinking, feeling, doubting part of you, was just more machinery. So he split reality in two. There was res extensa, the extended stuff, physical matter, everything you can touch and measure. And there was res cogitans, the thinking stuff, the mind, the soul. Two fundamentally different substances. This is Cartesian dualism, and it's one of the most influential ideas in Western philosophy. The mind is not the brain. The mind is something else entirely, something non-physical that somehow interacts with the brain. And here's the thing. Descartes even tried to specify where the interaction happened. He pointed to the pineal gland, that tiny structure sitting right in the middle of the brain, and said, "This is the seat of the soul. This is where the non-physical mind reaches in and steers the physical body." Why the pineal gland? Partly because it was one of the few brain structures that wasn't doubled. Most things in the brain come in pairs, left and right. But the pineal gland sits on the midline, singular, and Descartes figured the unified mind needed a singular point of contact. It's a beautiful idea in its own way. And it was wrong, of course. The pineal gland, as we now know, is mainly involved in producing melatonin and regulating your sleep-wake cycle, which is kind of poetic, actually, given that you're lying here right now and your own pineal gland is probably doing its thing, helping you drift off. But Descartes' real problem wasn't the pineal gland. It was the interaction itself. If the mind is truly non-physical, how does it push physical molecules around? How does a thought, which has no mass, no charge, no spatial location, cause your arm to lift? Philosophers call this the interaction problem, and nobody has ever solved it within a dualist framework. Not really. So over the centuries, as neuroscience accumulated more and more evidence that everything the mind does is correlated with something the brain does, most scientists quietly moved away from dualism. The working assumption became physicalism or materialism, the idea that the mind is what the brain does. Consciousness isn't a separate substance. It's a product of neural activity, the way a song is a product of vibrations in air. There's nothing extra, nothing floating above the machinery, just the machinery itself, doing something extraordinarily complex. But here's where it gets genuinely tricky, and this is important. Even if you accept that consciousness arises from the brain, and most neuroscientists do, you still have to explain how. And that turns out to be phenomenally hard. In 1995, the philosopher David Chalmers gave this difficulty a name that stuck. He called it the hard problem of consciousness. The easy problems, and he used the word easy, somewhat ironically, are things like explaining how the brain processes sensory information, how it integrates data, how it directs attention, how it controls behavior. Those are incredibly complex engineering challenges, sure, but they're the kind of thing you can imagine solving with enough detailed neuroscience. You trace the circuits, you map the computations, you build models. Hard, but tractable. The hard problem is different. The hard problem is, why is there subjective experience at all? When light hits your retina and signals travel through your visual cortex and you see the color red, why does redness feel like something? Why isn't it all just information processing in the dark with no inner experience attached? You could imagine a being that processes all the same information, produces all the same behaviors, but has no inner life whatsoever. Philosophers call this a zombie, not the horror movie kind, but a philosophical zombie, a being that's functionally identical to you, but has no conscious experience. The hard problem is explaining why you're not one of those. Why there's something it's like to be you. And this is where the honest answer from science is, we don't fully know. But that hasn't stopped some very smart people from trying, and the attempts are genuinely illuminating, even when they're incomplete. One of the most influential frameworks is called the global workspace theory, proposed by Bernard Baars in the late 1980s. Think of it this way. Your brain is doing an enormous amount of processing at any given moment. Your visual system is analyzing shapes and colors. Your auditory system is parsing sounds. Your motor system is maintaining your posture. Your emotional circuits are humming along in the background. Most of this processing is unconscious. You're not aware of the individual computations happening in each specialized module. What Baars proposed is that consciousness arises when information from these specialized processors gets broadcast to a kind of global workspace, a shared mental stage where different brain systems can all access the same information simultaneously. When something makes it onto that stage, you become conscious of it. When it doesn't, it stays unconscious. Think about driving a familiar route. Most of the time, the driving itself is handled by unconscious processors. You steer, you brake, you signal, all without really thinking about it. But then a deer jumps into the road, and suddenly that visual information gets broadcast to the global workspace. You're conscious of it. You feel the jolt of surprise. Multiple brain systems, attention, emotion, motor planning, decision-making, all get access to the same information at once, and you experience it as a unified moment of awareness. It's a compelling framework, and it maps reasonably well onto what we know about brain anatomy. There's evidence that the prefrontal cortex and certain long-range connections play a role in this kind of global broadcasting. When people are conscious of a stimulus versus when they're not. And you can set up clever experiments to test this, using things like visual masking, where you flash an image so briefly that sometimes people see it and sometimes they don't. The difference in brain activity often involves these widespread, synchronized patterns of neural firing that look a lot like what Bars described. But here's the honest critique. Global workspace theory is really good at explaining the functional side of consciousness, which information gets selected for broad access, how that selection works. What it doesn't fully explain is the subjective feel. It tells you what consciousness does, not why it feels like something. Another major contender is integrated information theory, or IIT, developed primarily by Giulio Tononi, a neuroscientist and psychiatrist at the University of Wisconsin. Tononi's approach is radically different. Instead of starting with the brain and asking what it does, he starts with consciousness itself and asks what properties it must have. And he identifies a key one: integration. Your conscious experience at any given moment is unified. You don't experience the color of the sunset separately from the warmth of the air, separately from the sound of the waves. It's all woven together into one seamless experience. At the same time, your experience is highly differentiated. Every conscious moment is unique, distinct from every other possible experience. Tononi proposed a mathematical measure called phi, which quantifies how much integrated information a system generates. The higher the phi, the more conscious the system is. And here's what's provocative about this. Phi is a property of the system's structure, not just its function. A system with lots of specialized parts that are also deeply interconnected, like the brain, would have high phi. A system where the parts operate independently, even if each part is very complex, would have low phi. And a simple feed-forward system, where information flows in only one direction with no loops or integration, would have essentially zero phi, regardless of how much information it processes. This leads to some surprising predictions. Under IIT, a digital computer running a perfect simulation of a brain would not necessarily be conscious, because its underlying physical architecture might not have the right kind of integrated information. Meanwhile, certain simple systems might have tiny amounts of consciousness, a controversial claim that edges toward panpsychism, the idea that consciousness is a fundamental feature of matter, present in some degree everywhere. Now, whether you find that beautiful or absurd probably depends on your philosophical temperament. And IIT has real critics. Some argue that phi is practically impossible to compute for any system as complex as the brain. Others challenge the foundational assumptions. But what's remarkable is that Tononi and his colleagues have been making testable predictions about what happens to consciousness during sleep, during anesthesia, in patients with brain damage. And some of those predictions have held up. When you're in dreamless deep sleep, for instance, the cortex loses much of its integrated activity. The neurons are still firing, but they're not talking to each other in the same coordinated way. The integration drops. And so, according to IIT, does consciousness, which matches your experience, or rather, your lack of experience, during deep sleep. And then there's a third approach that's been gaining traction, sometimes called predictive processing, or the Bayesian brain framework. This one comes from people like Carl Friston at University College London, and it reframes what the brain is fundamentally doing. Instead of passively receiving information from the world and building up a picture, the brain is constantly generating predictions about what's going to happen next, and then comparing those predictions against incoming sensory data. When the prediction matches reality, everything feels smooth and expected. When there's a mismatch, a prediction error, the brain updates its model. Under this framework, your conscious experience is essentially the brain's best guess about the world. You're not perceiving reality directly. You're perceiving a model of reality, a simulation that the brain constructs and constantly refines. Some researchers, like Anil Seth at the University of Sussex, have extended this idea to argue that consciousness is a kind of controlled hallucination. Not hallucination in the pathological sense, but in the sense that what you experience is generated from the inside, constrained and shaped by sensory input from the outside. When the system works well, your internal model matches external reality closely enough that you navigate the world successfully. When it breaks down, in dreams, in psychosis, in certain neurological conditions, the model drifts away from reality, and you experience things that aren't there. Think about what that means for right now. As you lie here, your brain is generating a model of your body's position, the texture of the sheets, the temperature of the room, the sound of this voice. None of that experience is happening out there. It's all being constructed in here, inside your skull, by networks of neurons making predictions and checking them against sensory data. And as you drift towards sleep, those checks get looser. The sensory input gets gated down. The predictions start running more freely, less constrained by reality. And that's when you start to dream. The model keeps generating experience, but without the anchor of external sensation to keep it honest. What's beautiful about these three frameworks, global workspace, integrated information theory, predictive processing, is that they're not necessarily mutually exclusive. They might be describing different aspects of the same underlying phenomenon. The global workspace tells you about access and selection. IIT tells you about the structural requirements for experience. Predictive processing tells you about the computational strategy the brain uses to build its model. Maybe consciousness requires all three. The right kind of integrated architecture, running a predictive model, with a mechanism for broadcasting selected information widely. But here's what nobody has fully cracked, and it's worth sitting with this for a moment. Even if we perfectly mapped every neuron, every synapse, every chemical signal in your brain, Even if we had a complete wiring diagram and a perfect computational model, would we understand why it feels like something to be you? Or would we just have an incredibly detailed description of the machinery, with the feeling itself still unexplained? Some philosophers think the hard problem will eventually dissolve as neuroscience advances, that it'll turn out to be a confusion born from our current ignorance. Others think it's a genuine, possibly permanent limit on what science can explain, and there's something almost comforting about that uncertainty, if you let it be. The fact that the most complex object we've ever encountered is the one sitting right behind your eyes, and it still holds mysteries that the best minds in the world are working on right now, at this very moment, in labs and lecture halls and late-night conversations all over the world. And maybe that's the right place to let the philosophy rest for a while, because the truth is, neuroscience hasn't been sitting around waiting for philosophers to settle the debate. It's been pushing forward, building new tools, and in the last couple of decades, those tools have started to change everything. Let's talk about what's happening now and where all of this is going. One of the biggest shifts in modern neuroscience started in the early 2000s with something called optogenetics. And it's one of those ideas that sounds like science fiction until you realize it actually works. The basic concept came together through the work of Carl Dieseroth at Stanford, along with Ed Boyden and others. And here's what they figured out. There are certain algae, simple, single-celled organisms that have light-sensitive proteins in their membranes. These proteins, called channel opsins, open up when you shine light on them and let ions flow through, which is essentially what neurons do when they fire. So the idea was, what if you could take the gene for that light-sensitive protein, insert it into specific neurons in a living brain, and then use tiny fiber optic cables to shine light directly on those neurons? You'd be able to turn individual groups of brain cells on or off with the flick of a laser. And it worked. By around 2005, they had demonstrated that you could control the behavior of living animals, mice initially, by activating or silencing precise populations of neurons with light. Think about what that means. For most of the history we've been talking about tonight, neuroscientists could observe the brain, or they could damage it and see what happened, or they could stimulate it with electricity, which is a pretty blunt instrument. But optogenetics gave them a scalpel where they'd previously had a hammer. You could target just the dopamine neurons in one specific circuit, or just the inhibitory interneurons in one layer of the cortex, and ask, what does this particular group of cells actually do? And the answers have been revelatory. Researchers have used optogenetics to implant false memories in mice, literally activating the hippocampal neurons that encoded one context, while the animal was in a completely different environment, creating an association that never actually happened. They've used it to instantly switch aggression on and off, to trigger feeding behavior in animals that aren't hungry, to silence the circuits involved in fear and watch an anxious mouse become calm in seconds. It's not that these findings are simple. The circuits involved are still staggeringly complex. But for the first time, neuroscience has a tool precise enough to test causal claims about what specific neurons do, rather than just correlating activity with behavior. And then there's the imaging revolution. You remember we talked about Penfield mapping the cortex with electrodes back in the 1940s and 50s? Well, functional MRI, fMRI, which became widely available in the 1990s, let researchers watch the whole brain in action without opening anyone's skull. It works by detecting changes in blood flow. When neurons in a region become more active, they need more oxygen, and the blood rushing to supply it creates a signal that the scanner can pick up. It's not perfect. The resolution is on the scale of millimeters, which means each little cube of signal, each voxel, contains something like 1 million neurons. And there's a time lag, because blood flow changes over seconds, while neurons fire in milliseconds. But it gave us something we'd never had before, a way to watch the living human brain think, remember, decide, feel, all in real time, or close to it. And here's what's really interesting. In the 2000s, researchers started combining fMRI with machine learning, training algorithms to decode brain activity patterns. A team at UC Berkeley, led by Jack Gallant, showed in 2011 that they could reconstruct rough visual images of what a person was watching just by reading their brain activity in the visual cortex. The reconstructions were blurry, dreamlike, but recognizably related to the original clips. You'd watch a movie, and the algorithm would produce a hazy approximation of what you were seeing, assembled entirely from your neural signals. That was over a decade ago. The reconstructions have gotten better since then. Other groups have decoded imagined speech, emotional states, even the gist of what someone is thinking about. Now, before you get too worried about mind reading, these techniques require the person to be lying still in a giant magnet, fully cooperating. And the decoding is trained on hours of that specific individual's data. Nobody is reading your thoughts from across the room. But the principle is profound. It means that the patterns of neural activity in your brain carry structured information about your subjective experience, and with the right tools, some of that information can be extracted. It's another piece of evidence that consciousness, whatever it is, leaves physical fingerprints in the brain. And this brings us to one of the most ambitious projects in the history of science. In 2013, both the United States and the European Union launched massive brain mapping initiatives. The U.S. effort, called the BRAIN Initiative, which stands for Brain Research Through Advancing Innovative Neurotechnologies, was announced by President Obama and has poured billions of dollars into developing new tools for recording and manipulating neural circuits. The European effort, the Human Brain Project, took a different approach, trying to simulate the brain computationally, building detailed digital models of neural circuits. The European project had a rocky start. It was criticized for being too top-down, too focused on simulation at the expense of basic research, and its leadership was restructured after a public revolt by hundreds of neuroscientists who signed an open letter in 2014. But both projects, in their different ways, reflected a shared conviction that neuroscience was ready to tackle the brain at a scale and resolution that previous generations could only dream about. And the results have been remarkable. We now have techniques that can record from thousands of neurons simultaneously in a living, behaving animal. A technology called neuropixels, a silicon probe thinner than a human hair, developed by a consortium led by Tim Harris at the Howard Hughes Medical Institute can record from hundreds of neurons at once across multiple brain regions. Teams at the Allen Institute in Seattle have been systematically cataloging every cell type in the mouse brain, creating an atlas of neural diversity that makes the old maps look like cave paintings. And in 2023, a complete connectome, a full wiring diagram of every neuron and every synapse, was published for a portion of the fruit fly brain, building on the earlier complete connectome of the roundworm C. elegans that was finished back in 1986 by Sidney Brenner's group. The fly connectome alone contained roughly 140,000 neurons and tens of millions of connections. The human brain, remember, has 86 billion neurons and somewhere around 100 trillion synapses. We are very, very far from a complete human connectome. But the trajectory is clear. So here's the question that all of this is building toward, and it's the question that connects everything we've talked about tonight. From those ancient Egyptians scooping out brains and throwing them away, to Broca holding a damaged hemisphere and realizing that language lived in a specific place, to Cajal squinting through his microscope at silver-stained neurons, to Kandel watching a sea slug learn. The question is, will understanding the brain's machinery ever fully explain the mind? And honestly, nobody knows. But here's what I find genuinely moving about the current moment. We are the first generation of humans that can even ask that question with the right tools in hand. Every generation before us was guessing, brilliantly in many cases, but guessing. We are starting to actually see, and the practical implications are already arriving. Deep brain stimulation, surgically implanted electrodes that deliver precisely targeted electrical pulses, is now an established treatment for Parkinson's disease when medication isn't enough, and it's being explored for treatment-resistant depression, obsessive-compulsive disorder, and chronic pain. Brain-computer interfaces, the kind being developed by groups like BrainGate and by companies you've probably heard of, have allowed paralyzed patients to control computer cursors, robotic arms, even to type messages, just by thinking about the movements. In 2021, a man who'd lost the ability to speak after a stroke had an electrode array implanted on his motor cortex, and researchers at UCSF decoded his attempted speech at a rate of about 15 words per minute. It was slow, halting, imperfect, but he was speaking through a machine, using his thoughts, for the first time in years. Think about what that means for someone trapped inside a body that won't cooperate. Think about what it will mean when the technology gets better, which it will. And yet, for all of this progress, the brain keeps humbling us. We still don't fully understand how general anesthesia works. We use it every day in hospitals around the world, and we know it suppresses consciousness. But the precise mechanism by which certain molecules make awareness disappear and then come back is still debated. We don't understand why we need sleep. Not really. Not at the deepest level. We know it's essential. We know terrible things happen without it. We know the brain does important housekeeping during sleep, clearing metabolic waste through the glymphatic system, consolidating memories as we discussed with HM. But why evolution chose to make every complex organism periodically lose consciousness and become vulnerable to predators for hours at a time? that's still an open question. We don't understand how the brain generates the seamless, unified experience of being a self, how billions of neurons, each doing their own tiny electrochemical thing, somehow produce the feeling of being one person, with one perspective, one continuous story. And maybe that's okay. Maybe that's more than okay. Because the history of neuroscience, the whole long story we've been walking through tonight, is a history of people staring at the most complicated thing in the known universe and refusing to look away. Every generation understood a little more than the one before. Every wrong turn, the animal spirits in the ventricles, the bumps on the skull, the idea that the brain couldn't change. Every wrong turn eventually led someone to the right question. And the right question, asked at the right time, with the right tools, changed everything. So here you are, lying in the dark, and your brain is doing all of the things we've talked about. Your hippocampus is quietly sorting through the day, deciding what to keep and what to let go. Your cortex is running its predictive models with the sensory gates turned down, which is why the darkness behind your eyelids might be starting to shimmer with the beginnings of something, a scene, a face, a place that doesn't quite exist. Your neurotransmitters are shifting their balance, serotonin and norepinephrine fading as the sleep systems take over, GABA quieting the networks that kept you alert and focused during the day. Your synapses, the ones that were strengthened by whatever you learned and experienced today, are being consolidated, the important connections reinforced, the trivial ones pruned back. You are, right now, a living demonstration of everything we've been talking about. And the thing that gets me, the thing I keep coming back to, is that the organ doing all of this work is the same organ that's trying to understand itself. That's the great recursive puzzle at the heart of neuroscience. The brain is both the instrument and the object of study. It's as if a telescope were trying to turn around and look at its own lens, or a hand were trying to pick itself up. There's something almost paradoxical about it. And yet, here we are, making progress anyway. Slow, imperfect, beautiful progress. The mysteries that remain aren't a failure. They're an invitation. And the story of the brain, your brain, the one that's been listening to all of this, the one that's getting heavier and quieter now. That story is still being written. By researchers in labs you'll never visit, by patients whose courage makes the science possible, and by the sheer stubborn curiosity of a species that looked at the wrinkled grayish lump inside its own skull and thought, I want to understand that. I need to understand that. So let it rest now. Let the hippocampus do its work. Let the predictions run free and the dreams come if they want to. You've been carrying around the most extraordinary object in the known universe your entire life, and it's been quietly keeping you alive, keeping you curious, keeping you you, every single second. It doesn't need your conscious attention to do what it does next. It's been doing this since before you were born. Trust it. Let it carry you the rest of the way into sleep. And if any of these stories stay with you, Kajal and his drawings, Loewi and his frog hearts at 3 a.m, the sea slug learning to flinch, the patient who spoke through a machine for the first time in years, if any of it lingers, that's your synapses deciding it matters. That's long-term potentiation, doing exactly what Heb predicted it would. That's your brain, rewriting itself, one connection at a time, in the dark, while you sleep.